Good morning. My name is Kalyan Banda. I'm a medical oncologist at Seattle at the University of Washington and the Fred Hutchinson Cancer Research Center. I specialize in gynecological malignancies and experimental therapeutics, and thank you for having me this morning. Today, we're going to talk about what are targeted therapies and immunotherapies. This talk is not meant to be a comprehensive overview or provide serious detail. The goal of this talk is to provide a brief and overall understanding of what are these therapies, why do they work, and some situations where they would be used. And to go in, I do get research funding from Eli Lilly. I am their site PI for a trial that has no bearing on the talk today. Before we understand what targeted therapies are, we need to understand what a cancer is. I mean, the answer should be obvious enough, but the real question I'm asking is, what is unique about a cancer cell compared to a normal cell? And about 20 years ago, two world famous scientists actually put that list together, and they listed out what they called hallmarks of cancer, something that are unique in driving cancer compared to a normal cell. And these is a visual representation of some of them. So let's talk about what are these unique abnormalities. Fundamentally, cancer is an outlaw organ. It's an organ that refuses to be confined to its niche, which means it pokes through into neighboring tissue, what's called invasion, or goes to a site that is not supposed to be, what's called metastasis. So it's not confined to the normal place. Second, it's something that escapes the normal guardians of your genome. For example, P53 is a gene that regulates all of your genes in the sense that anytime that there's a catastrophic damage to other genes, P53 shuts down the cell and makes sure that it self-destructs. If there's a mutation in P53, which can be inherited through Li from any or acquired in the cancer itself, this checkpoint, this guardian is suddenly vanished. And the cancer and the cell can now escape the normal mechanism and start multiplying. Which brings me to the third hallmark, which is the sustained and relentless drive to grow. It just keeps growing with no off button. And at the same time that it's growing, it also has learned how to escape cell death, something that every cell should normally do. Not only does it escape death, it then grows its own blood vessels, ensure its own nutrition, its own blood supply, and can keep growing. And while it does that, it also multiplies endlessly. One cell becomes 2 cells, becomes 4 cells, becomes 8 cells, and so on and so forth, a characteristic that normal cells in our body should not have. So 10 years ago, these hallmarks were updated to include new ones. Importantly, escaping the immune system. One of the fundamental abilities of cancer is to tell our immune system that I'm normal, I'm part of you, do not attack me, and thus escape death. At the same time, they also enhance their own metabolism, figure out a way to continuously feed themselves while they grow, they proliferate, they don't die. All of these hallmarks occur in the background of two enabling characteristics or enablers. These are the things that drive the hallmarks. One, genomic instability or mutation. Most famous example is an inherited BRCA1 or 2 mutation that causes widespread instability or mutations in the cancer, similar Lynch syndrome, or environmental factors like UV that constantly damage your DNA. At the same time, there has to be some constant background of chronic inflammation. It could be environmentally induced, such as smoking for 50 years, or it could be something like ulcerative colitis, where there's constant inflammation of the colon that creates a field ready for cancer to develop in. So all cancers have these hallmarks and these enablers, all 8 hallmarks and 2 enablers. What's different from each cancer to cancer is the relative importance of each hallmark or enabler. Now that we know what's unique about these cancers, we've also provided an opportunity to target these unique abnormalities. And that, in a sense, is what targeted therapy is. There are things that exploit these unique biological abnormalities in cancer and target them specifically. Contrast this to chemotherapy, where chemotherapy kills anything that grows quickly, whether it's normal or abnormal. But the difference is the normal cells that we have redundancies built in and have the capability of recovering from these insults, whereas you're hoping the cancer cell does not recover from these insults. And that's the difference between chemotherapy and a targeted therapy. But targeted therapies also lead us to this modern concept that hallmarks have become targets. And here are a few listed examples of how we've turned hallmarks today into targets, pills that are available today in many cancers. Again, famously, PARP inhibitors that target genomic instability or mutations, EGFR inhibitors in lung cancer that have turned off this relentless drive to keep growing, or immunotherapy, which I'll come to later, that have taken away this hallmark of avoiding immune destruction. And these now pills and these drugs have turned modern oncology on its head. And the new paradigm is what we call precision oncology, where you're trying to tailor the medical treatment to the individual characteristics of each patient, target those unique vulnerabilities of each individual's cancer. So how do we do this? And why, in a sense, do targeted therapies work? I already told you they work because they have these hallmarks, but you can't just target a random hallmark because all of them exist in all cancers and hope that they work. There have to be something unique that makes this targeted therapy work. And there are two situations that need to occur. One, you need to figure out what this addiction that the cancer has. Each unique cancer has an addiction. It's dependent on a few genes or pathways or biological abnormalities to keep growing and maintaining itself. These are different for each cancer. And one famous example is what we call HER2/neu in breast cancer. If this is sitting on top of the cancer, it just drives the cancer relentlessly to grow, to multiply. And this is the dominant driving force of breast cancer when it's present. Very similar to HER2/neu is EGFR. For example, in lung cancer, you can have a mutation of this thing sitting on top of the cancer, and that mutation results in it being turned on all the time. There is no off button anymore. Now the cancer just goes on and on and on like the ever-ready bunny. So all you need to do is take off this battery and you've targeted the Achilles heel of a cancer. And that, in a sense, is one of those situations where targeted therapy work. And so we do, before we start using targeted therapy, have to figure out what addiction does this particular cancer have in this particular situation. Apart from figuring out the addiction that the cancer has, what we call oncogene addiction, we have to figure out a second situation, something what we call synthetic lethality, meaning we have to figure out a situation and a drug where the cell death occurs only in the cancer and the normal cells are spared. And I'll give you an example. The best example here is in BRCA mutations. BRCA mutations result in a defect in DNA repair called homologous recombination deficiency. If you don't have the mutation, you don't have the defect. If you have a mutation and you have a normal cell, you don't have the defect because you still have a normal copy, functional copy of BRCA. But the cancer has no BRCA that's functional and has the homologous recombination defect. But the cancer can still survive because it reroutes DNA repair through some other pathways that are called like base excision repair. Now you give them a PARP inhibitor, that PARP inhibitor shuts down the rerouting pathway, the base excision repair. Now the cancer has no homologous repair, has no base excision repair, and this A plus B results in the cancer cell death, but not in normal cell death. And this is what you need to find for a targeted therapy to be effective and to be safe. You need to find the addiction, you need to find the synthetic lethality. And that's how modern targeted therapies work. So let's talk about a famous example. Chronic myeloid leukemia is the original poster child. It is caused by a single genetic abnormality, what we call a genetic translocation. Two parts of different chromosomes have mixed and matched to create this crazy chimeric child that relentlessly drives cancer. So this cancer is a single abnormality. That single abnormality can be targeted by a single drug unique to that abnormality, which means it doesn't affect normal cells. And with that drug, we've converted chronic myeloid leukemia from a deadly disease to a chronic disease. Naturally, when this came out decades ago, it became a Time magazine cover. This was considered a breakthrough, the magic bullet against cancer. A pill a day keeps the cancer away. That was the new mantra. And we thought, this is how all of cancer will be, that we just need to find that one single abnormality for each unique cancer and target it with this one pill and it'll take care of everything. Today, we know better. That's not the modern scenario. The modern scenario is Each cancer is actually a dozen different kinds of cancers at a genetic level. And for each kind, you just need to find that abnormality and target it with a unique pill. For example, lung cancer. Lung cancer at its genetic level is probably several dozen different kinds of cancer. And each maybe a small percentage of the general like lung cancers that exist, but each can be targeted by a different kind of pill. And by doing this, we're attacking each different kind, but it's by its own vulnerability and changing, hopefully, lung cancer from an incurable disease to a chronic disease. That is the promise of targeted therapy and precision oncology. So let's talk about just a few examples. PARP inhibitors in the presence of BRCA1 and 2 mutations. There are several different kinds of PARP inhibitors, and they can be used in many different cancers where there are BRCA1 and 2 mutations: breast, ovarian, pancreatic, prostate. Most commonly, these are inherited germline mutations, meaning you're born or you inherit these mutations, and therefore there is this unique vulnerability. Many times, it is not inherited. The cancer itself can acquire a mutation BRCA1 or 2 sometime during its development, but that still leaves a unique situation that can be exploited. And with this, usually these PARP inhibitors are used in advanced, incurable, or metastatic cancers. And you're hoping that by taking this pill, you're changing that incurable cancer to a chronic disease, at least for a while. The other situation now we're using them is in early cancers, where the hope is that you can take this early cancer, treat them with surgery, chemo, all of those things, but then add these PARP inhibitors on and convert early stage cancers to fully curable cancers. Make sure they never come back again. Other famous examples are like PIK3 inhibitors in breast cancer, alpelisib. PIK3 mutations, these kind of mutations are typically not inherited, they're not germline, but they are acquired or somatic. But that still gives us this unique opportunity to target it. And for example, FLT3 inhibitors in AML are definitely not germline or inherited. They're almost always acquired, but they are what drives the cancer and therefore can be targeted. Which does bring me to my point that targeted therapies are unique to a cancer's vulnerability, whether it's inherited or acquired. And many patients with germline mutations have other additional acquired vulnerabilities in their cancer that must be targeted. So how would you know whether to target this or not? Currently today, the best way to know is what we call genetic sequencing. There is the inherited sequencing, which is separate. I'm not talking about that. I'm talking about sequencing of the cancer to find out what are the unique vulnerabilities of this individual specific cancer, and can we target it with these particular pills? And that, in a sense, is targeted therapy today. Which brings me to the next part of the talk. What are immunotherapies? They are treatments that then alter an individual's immune system to kill their cancer. That is the difference between targeted therapies, immunotherapies, and chemotherapy. The immunotherapy in itself does not usually kill cancer, but it is harnessing the power of an individual's immune system to kill that cancer. Why would it work? I already told you that evading the immune system is a hallmark of a cancer. All of us have an immune system that develops usually when we are fetuses. And what the first thing it learns is what is self and what's not self. What defines this individual, what defines not this individual. And cancer is a part of us. It does come from our own normal cells. It has several abnormalities. What it can do, in a sense, is then keep telling our immune system that I'm normal. Our immune system indulges in handshakes all day. That's how we as human beings tell another human being that, we're safe. I'm your friend. I'm not your enemy, right? We smile, we give them a handshake and say, hey, how's it going? That's the first indication that this individual is not a threat. That's also what the immune system does. It constantly tries to, do handshakes and smiles with all the cells that it encounters. And cancer has figured out a way where it does this handshake, it smiles and say, I am not a threat. I am part of you. And there are these particular things where proteins that meet together to do this handshake. Now, the beauty of immunotherapy is that you block these handshakes. The most common way to do this is through monoclonal antibodies like pembrolizumab or Keytruda, nivolumab or Opdivo, ipilimumab or Yervoy. What they do is interrupt this handshake. Once the handshake is interrupted, you've taken the brakes of your immune system. Your immune system is now able to recognize the cancer as abnormal and start chomping at it. And that's how immunotherapies work. They really work best if your cancers look quite abnormal, meaning the cancer looks quite different from a normal cell. And that is when the cancer has several mutations and is constantly changing. Those kind of cancers are usually melanomas or lung cancer or bladder cancers. For some reason, these cancers just have a lot of mutations. Otherwise, immunotherapies have to be combined with other chemos in some situations. But there are some other situations where immunotherapies by themselves have magical responses. And that is something that we call deficient mismatch repair or microsatellite instability. There are certain genes called mismatch repair genes. They are the ones that repair your DNA when it's damaged through a particular pathway. If there are inherited mutations in some of these mismatch repair genes, that is Lynch syndrome. But apart from Lynch syndrome, the cancer itself can acquire mutations in some of these genes and develop a mini Lynch syndrome within the cancer itself. And that is the situation where immunotherapy can work best. A couple of years ago, FDA granted approval for pembrolizumab in the presence of a mismatch repair or microsatellite instability, regardless of what kind of cancer it is. This is the first time that FDA did something like this. Usually what FDA does is it approves a drug for a particular type of cancer, for a particular stage of cancer. It doesn't say, we'll approve the drug, just use it in whatever cancer you want. But this is also how modern oncology is changing. Is that we recognize that some abnormalities are common to cancers. Not all cancers, but some cancers. And exploiting those abnormalities across cancers will result in benefit. And so pembrolizumab is one that works here. And as a reminder, mismatch repair can be inherited or non-inherited. It can be across different kinds of cancer, and it will result in great responses to pembrolizumab, which means we must use it. How would you know you have it? Again, through sequencing. Lynch syndrome is germline, so it's germline sequencing, and mismatch repair is through the, microsatellite instability is when you sequence the cancer itself and find it. And we do it. Some cancers is very common, like colorectal endometrial cancer. Some cancers it's very rare, like breast cancer, but you still do it because the benefit, if present, is huge. Now, immunotherapy has gone beyond these monoclonal antibodies. Nowadays, we talk about things like adoptive T cell therapies. The common terms you may hear are CAR T cell therapies or tumor-infiltrating lymphocytes or TIL therapies. And the basic strategy here is you're not just taking the brakes of your immune system, you're actually harvesting part of your immune system, growing it out or modifying it or expanding it and giving it back to this individual. CAR T cells, the way it work is you harvest a cell, you re-engineer it so it can target something specific to the cancer and not normal cell. You grow it out so there are millions of these cells and you give it back to the individual. These are already FDA approved for some leukemias, some lymphomas, some multiple myelomas, and there are dozens and dozens of clinical trials otherwise going on in cancers. Now, TILs are a little different, but the same basic concept. So this is one way we do immunotherapy. Now, the last way we're doing immunotherapy are truly vaccines. People have tried to do vaccines for a long time. We've just now gotten very good at it, as you might know from this thing we call the COVID-19, and it's vaccine. What you're trying to do is just like you find a unique protein that's in the influenza virus or COVID virus, and then you teach your immune system to react to it, the same thing, but you're doing against a cancer protein. Find a unique protein that exists only in the cancer, give it as a vaccine to the individual, maybe give it to them repeatedly so that the immune system learns that this is abnormal and then starts attacking the immune system. Again, there are lots of clinical trials going on with cancer vaccines, and this is a active area of research. So what is the promise of targeted therapies and immunotherapies? It is this modern concept that we can no longer be carpet bombing cancer. We have to target an individual's and an individual cancer's unique vulnerabilities. We need to change oncology from something that's just a cookie cutter approach to something that's highly targeted and precise. These therapies are not restricted to germline mutations or syndromes, but they may work best in some germline mutations or syndromes. So every individual with a germline mutation or syndrome must still have somatic sequencing of their cancer because they may have additional vulnerabilities that can be targeted. And the great hope today is that we can take all incurable cancers and make them chronic diseases that are mere nuisances. Take it from something deadly to something that will not affect the quality or quantity of an individual's life. The other promise that we take all early stage cancers and treat them so effectively that they will never come back again. This is the promise of precision medicine. It still has a way to go to achieve that promise, obviously. How are some ways that you might benefit if you are an individual with cancer? Please talk to your physician about any targeted therapies or immunotherapies that are available for your cancer. Second most important thing is please consider clinical trials. All of these therapies today have come from clinical trials. And those trials have direct benefit to an individual. They also have larger benefit to society. So please consider how and when you can participate in a clinical trial. Beyond that, I hope you have a great day. And I thank you for this opportunity. I'm sorry, but I will not be there to take questions because I am giving another talk at the exact same time. Again, have a great day and thank you again.